individual letters of the DNA code. Those are our bases. And we can actually divide up those bases into two different types. Now, if you'll look real quickly, you'll see at the top that all bases have certain things in common. They all, because this is DNA, they have deoxyribo. Everybody see in the picture right there, we have our bases forming. Deoxyribose, we have a phosphate, and then we have a base. Everybody see the three structure part of that? So and on that the top line of the picture, right, we have a base attached to a deoxyribose, then we attach that to a phosphate. That's what each one's going to look like. It's going to have a phosphate, it's going to have a sugar, that's the pentagon, and then it's going to have a base. And the base is going to be what's important, okay? The base tells us what exactly we have right there. And the bases, which we've already learned, those are our ACTG, and then in RNA we replace T with U, okay? They can be divided into two different groups based on their structure and the way that they work, okay? So the first group is called the purines. I know it's flipped in the picture in my notes. I meant to go back and fix that, but I didn't. I want you to find the pictures of the purines. Everybody see the purines? Mm-hmm. Okay, what are our two types of purines?
Adenine and guanine.
Adenine and guanine. Now, how many rings do the purines have?
Two.
Two rings, right? They have two rings. And by ring, I just mean a ring, a structure that goes around. So they're going to be the big bases, right? They're going to be the big bases. The purines are the two-ring bases. They're going to be larger. Now, for reasons we don't necessarily have to get into, I'll tell you this right now. Adenine likes to form two hydrogen bonds. It likes to form two hydrogen bonds. Guanine likes to form three hydrogen bonds. Okay? Adenine likes to form two, guanine likes to form three. Are they going to fit together well? No. Adenine and guanine do not stick together. They will not stick to each other. Okay? And that's a good thing because they're too big. If they stuck together, we couldn't form our ladder because the ladder would be too fat in certain parts. Okay? So, the purines will have to attach onto what?
The
Pyrimidines. The pyrimidines, which if you look, how many rings does each pyrimidine have?
One.
One. So they're the short one. Okay? So each purine will attach to a pyrimidine, which has only one ring.
Wait, what is the pyrimidine?
So, with the same picture, right? I know they're flipped from the way I've got them in the notes, but you can see them, right?
Okay, so what likes the
So purines are going to like a pyrimidine,
okay.
right? And that way, they're going to bond, so a fat one with a skinny one, right? That's how they like to form. Now, do you remember in DNA, what does adenine like? Which one was adenine attached to? Thymine, to thymine. So if adenine wants two hydrogen bonds, how many bonds must thymine want? Two, okay? So thymine will want two bonds. Now, who remembers what replaces thymine in RNA? Do you remember we actually... Uracil. So what must uracil also want?
Two.
Two. Otherwise, it's a bad replacement, right? It has to be pretty much just like thymine to work, and it is.
Wait, how many did cytosine?
Okay, we'll get that in a second, right? Adenine wanted two, so it'll bond to thymine and uracil, who also both want two, okay? Now, how many did we say guanine wanted?
Three.
And who does guanine like to bond to?
Cytosine.
So therefore, cytosine must also want
Three.
three hydrogen bonds, okay? And this is actually, it's that simple. This is how all DNA works. Adenine likes thymine because it has two hands to hold hands with. And so it'll only hold hands with other two-handed bases. In fact, it won't hold hands with itself because it'll be too fat, right? So A, even though it likes to have two bonds, won't hold on with another A because it would be too fat. It will only hold on to a purine who wants two bonds, which is thymine in the nucleus, and outside the nucleus, uracil for the RNA. Isabel?
What did you say a base was? Like,
So a base is the individual letters, the individual units of a nucleic acid, right? So DNA is made up of the individual bases, of which in DNA there's four types: two purines, two pyrimidines. Okay? And this will dictate the way they function, the way they join together. Okay. Now, this next part, now that we know how DNA sticks together, we can see pretty easily how to make more of that DNA. And this is DNA replication. Does anyone remember when DNA copies. Not when the cell copies, but when does DNA copy?
Reproduction.
Telophase?
It's actually not, it's not during
Interphase.
mitosis.
Synthesis?
What?
Synthesis?
And when does that synthesis phase happens when?
Before.
And what we call the before mitosis? Interphase. It happens during the S phase of interphase. So this replication will happen in the S phase of interphase. Remember, before we can divide, we already need a double amount of DNA or division won't work. Our cells won't have enough DNA. So this will happen in the S phase of interphase. We're going to copy our DNA, and that's what DNA replication is. And if everything goes smoothly, we should get two identical ladders, two identical double helixes, right? Does everything always go smoothly? No, but usually, because DNA is really good at checking itself, DNA is only in like 1 in, I think, about 10 million times will it actually go wrong, which is really rare, but you have about 1 trillion cells in you, right? And so there's actually tons of cells this happens to, right? So it actually happens all the time, even though it's super rare. 1 in 10 million is super rare, unless you're talking about an organism that has 1 trillion cells, in which case it happens pretty much every minute. But usually you fix it or you kill that cell, so it's not a problem. So almost never does it really cause a serious problem. Okay, so we're going to go through the process of replication. I'm going to draw a little bit, but if you'd rather, there's actually a pretty decent picture in your book. If you will look on page 292 and 293. And you can actually see in the picture, if you look at an A, how many hexagons does A have in that picture on 292 and 293? You'd have to look at it.
How many what?
So I want you to see the little A, the green A. How many little polygons are there? Little hexagons are stuck to each other. And look at the T, how many are in that? Right, because a pyrimidine has 1, a purine has 2. All right, that makes sense to everybody in the picture? Oh, yes,
The
ma'am.
hexagons are rings,
Yes, they're rings. They're just filled in rings here.
and that doesn't have to do with the bonds. How many bonds it has.
Well, how many bonds it has is actually, if you look at your notes,
That-
it has to do with how many hydrogens are available. But we're not worried about it. You just need to know that adenine and thymine want 2, cytosine and guanine want 3. That's why they like each other.
Why is one of the rings like bigger than the other? Like, is that just how they draw it?
One's a hexagon, one's a pentagon.
So like in like real life, are they the same size?
No, they're like that. This is pretty accurate how they really look.
Why are they different sizes?
Different shapes. And
What do you-
different shapes mean it actually has a lot to do with how many bonds they are.
Okay.
Okay, so first things first, if we have a ladder and we want to copy the ladder, what do we have to do?
Split it.
We got to split. We got to pull it apart, right? Remember, because the words are on the inside. So I'm going to have to split it apart, and we call this separation. And separation works not exactly, but almost exactly like a zipper works, right? Zippers are linked together, and then when you run a zipper, I don't know if you've ever looked at this, but one side has a wedge, and the other side has kind of a fat part. Have you ever noticed that before? And the wedge is a funny one in that it actually, as it goes through, it pushes one part down and one part up, and so it detaches each part of the zipper. Well, all that's holding together the DNA in the middle is hydrogen bonds. What do you remember about hydrogen bonds? They're weak. They're easy to and easy to make, right? Easy to break, easy to make. So we don't really even need a lot of energy. We just need a zipper. And this is going to be an enzyme whose name we're not too worried about. It's going to zip up the DNA, and as it goes along, it's going to split apart each strand, okay? So separation, we have our enzyme acting like a zipper, splitting apart each strand of the DNA. And when we do this, we will have, I'm just going to focus on one strand, well, that one's not working. We'll focus on one strand, one side of the separated DNA. So if it has, let's say, A, T, A, C, G, G, now they're going to be lying out here by themselves. But how many hands is adenine holding out?
2,
How many is thymine?
2, 3, 3.
Right? Then they're just holding out these hands, ready to grab on with somebody. They're lonely, lonely bases. Now, if we take a bucket of free nucleotides, so if we take a whole jar full of all of these nucleotides, and by the way, where did we get these nucleotides?
Eat it.
We ate them, that's right, we ate the DNA of other things. And if I just dump this out onto here, who's going to stick right here?
A T.
Who's going to stick right here? Who's going to stick right here? Right here? Right here?
C.
Okay, in fact, and they will stick there. I don't even have to put them on there. I just have to throw them onto here. They will order themselves perfectly for me because of the number of hands they like to hold.
Well, I want an A stick to an A. Whatever.
Ah, because if you remember, an A is shaped like this with two hands. And so if it's shaped like this, it'll be much fatter than all the rest of them who are only 3 rings wide. Okay? And so what happens is when our enzyme comes back over this, it says, whoa, that's wrong, and it kicks this guy out. And then if another A shows up, it'll kick it out too.
Is it T is small?
And the T is going to be small and fit in there. Does that make sense?
Is it ever possible for thymine and uracil to be present at the same time?
Uracil is not usually allowed into the nucleus, and so and thymine's kept in the nucleus.
So
But it can't. I mean,
will
this
they
thing
stick
happens
together?
100 billion times a minute in your body. So anything that can go wrong
Will they
goes
stick
wrong.
together since they both?
Well, they won't stick to each other because they're both purines, but either one will stick to an A.
Oh, okay.
Every now and then, in fact, this is why the sun is so bad for you. If you have 2 T's right here, and they're next to each other, they will actually, if the sun blasts apart the A's on the other side, which is what it does, it breaks these bonds, they will sometimes grab onto each other, and that's actually one of the leading causes of skin cancer. If that happens in the melanin gene, or in a basal cell, that actually causes one type of basal cell carcinoma. It's called thymine dimer, and it messes everything up.
I don't understand what uracil is.
So uracil is thymine for RNA.
Okay, so it only shows up in RNA.
Only in RNA. That's why I'm not drawing any U's, because where are we?
DNA.
We're in DNA. Does that make sense?
Mhm.
So in fact, we're going to get to RNA on the next page, and then we'll ignore T. There'll be no T's.
Okay.
Okay, so now if you notice though, right here, I've now put these together. What do we call when we stick these bases together?
Base pairing.
Base pairing, right? I have paired the bases. But if you notice something, in my ladder, I only have a rope on one side. Now, how well will a ladder work with a rope on only one side? It doesn't really work, right? So what am I going to have to do?
Add another strand.
Right, I'm going to actually, and each one of these actually have their own strand like this. But what do I need to do? I need to stick them all together. And this is my bonding, right? This is my bonding all the bases together. And now, what have I made?
A ladder.
Yeah. And what's on this side?
More.
Well, yeah, but also, because remember, up here I have the corresponding strand going T, A, T, so on and so forth. And it's going to make one just like this over here.
I don't get that.
Okay, so up here,
Oh, because you split it apart.
uh-huh.
Oh, I get it.
So when I split these apart, that's going to make a new one, that's going to make a new one. So how many total will I get once I've done all this? 2. And I've replicated my DNA. Isabel?
Wait, so where did you get the line that attaches to it to make it DNA?
Well, so they actually, my T really looks like this, right? And so when they paired up, they were like that. And then all I need to do is go stick them all together.
So what does that? What does
An
it just do?
enzyme.
Okay.
And enzymes we're not worried about. Basically, anytime a step happens here, there's an enzyme coming in.
Okay.
Okay? Okay, so that's DNA. I know it was quick. Any questions on that? We will be going very quickly. Can I not wait?
What was the bonding of bases? That was when they stick together?
That's when we form our pair rope. So the bond of the bases. Remember, we paired them all. Now we just have to attach them together. Make our other rope. Which we do, by the way, by taking away water. What do we call that?
Hydrolysis.
Other one.
Wait.
Dehydration synthesis, right?
Yeah,
Because see, we're
okay.
making bonds. If we wanted to break it, we'd hydrolyze. Does that make sense to everybody? So to make bonds, we dehydrate. Dehydration synthesis.
So you make it a ladder by drinking.
So right here we pull out the water, they stick together. Pull out the water, they stick together. Pull out water, they stick together. So we pull out the water, form
Where
a
did the
bond.
oxygen come from?
What if it's already in there? That's why we took out the water, the H2O.
Yeah, so like where did they get it?
Not to go into too much detail, but this phosphate will actually have an O on it right here.
Isn't that a fat?
A phosphate? No, that's a phospholipid.
Uh,
Then we'll have two hydrogens, and we'll pull them out, and then this guy will attach to the sugar because water's gone, and it has no one to hold hands with, so it holds hands with the sugar.
that's sweet.
Adorable.
Because
Okay,
it's
yeah,
sugar.
gotcha. Thanks. Okay, so let's take a look. Any questions on this? I don't want to raise it yet. Let's take a look. Probably if you didn't write that down, there is a pretty good picture of it on page 292. So if you want to write page 292 on your notes there. Okay. Let's take a look at RNA next. Now, if DNA was deoxyribonucleic acid, what will RNA be? Ribonucleic acid. In other words, so deoxy means without an oxygen. So ribonucleic acid will be almost the same thing, but it's going to have one more oxygen, right? Ribonucleic acid is going to have one more oxygen. Now, unlike DNA, in fact, having another oxygen actually makes it not twist, so it can't actually be a spiral ladder because that oxygen kind of gets in the way. It repels something too strongly. So we can't actually twist with this. So who remembers what shape RNA is?
It's like half.
That's right, it's just one strand, right? It's just one strand. RNA is a single strand. What's some advantage to being just one strand?
Easy to
Easy to move.
move.
It's also easy to,
You can do make.
you can make more shapes with it, that's actually true. But also, how expensive is it?
Not very.
In fact, it's exactly how much expensive? Half, right? One strand is half as expensive because, you know, we have to gather all these things up. We have to get the resources for it. It's half as expensive. What's the problem with it? What's the downside to it?
Easy to break.
True.
Also easy to misread.
And that's right, it's easy to mess up, right? We have no second editor. So RNA mistakes are much more common. I think it may be something like 1 in 100 times we get an RNA mistake as opposed to 1 in 10 million for DNA because of its checking mechanism. But that's okay. When we make a bad RNA, we just eat it up and make another one and try again. So RNA does have more mistakes, but it's much more mobile. And it's cheaper. So it's going to be better for doing jobs other than storing information, right? For storing information, we're okay with it being bulky because it needs to stay the same. But if we're going to be doing other things, we want something we can use that's mobile. So it's going to be a single strand, and we're going to be able to fold it into a whole bunch of different shapes. There's one other difference from DNA. What happens to thymine?
It turns into uracil.
Yeah, it gets replaced by uracil. It doesn't actually turn into it, but it is replaced by uracil.
What is?
So thymine gets replaced by uracil. So instead of ACTG, what will our 4 letters be in RNA?
AUCG.
AUCG. AUCG, right? Those are going to be our RNA letters. Okay. Now, whereas in DNA, the only thing we can do with DNA is have it be a ladder. Can't do a whole lot with it. I don't know if you've ever tried this, but it's pretty hard to make stuff with a rope ladder. It's pretty hard to tie a rope ladder into a knot, right? Or make, say, balloon animals with it. It's a pretty good shape at being a ladder and being stable, but that's about it. Okay? Single strand, we can do all kinds of stuff. We can tie it into knots, we can wad it up. We can do all kinds of things with it. So because of that, we're going to have a bunch of different types of RNA, whereas we only had one type of DNA. Okay? And each one will get named by a prefix that tells us what it does. Each type of RNA will be named with a prefix telling us what it does. Our first RNA is mRNA. The M stands for messenger. It is a messenger RNA.
Wait, is that like?
That's exactly what it is. No, that is what the M stands for. The M stands for messenger. The people who discovered this were Americans in the 1960s. They saw no reason to give it fancy names.
That's good.
God bless America.
So, messenger RNA is going to be exactly that. It's a messenger. What do messengers do?
Message things.
What do they take though?
Like parcels.
Information, right? A messenger is not going to deliver things. That's a delivery man. A messenger will carry information somewhere, right? Now, where's the information stored?
In the
In the nucleus. It's DNA in the nucleus, right? So we're going to travel from the nucleus. But here's the thing, what do all these messages mean? What does protein code for?
Like how to build.
I mean, what does it mean, what does DNA code for?
Proteins.
Proteins, right? So, we're gonna have to take it from the DNA to where we make proteins. Who remembers where we make proteins in a cell?
In the
In the ribosomes, that's right. So, where's this messenger gonna go? From
The
The
nucleus.
From
nucleus.
the nucleus to the ribosomes. That's where this messenger goes. He travels from the nucleus to the ribosomes with the instructions on how to make one protein, right? Instructions on how to make one protein. He's the messenger. And because he's just a messenger, he's going to be a long, simple strand. Okay, mRNA is just a long strand, it's just a letter. It's not even sealed. There's no worries about mail being stolen here.
Normal size.
Okay. And it's going to go to the ribosome. Our next part of RNA is going to be tRNA. The T stands for transport. It is the transport RNA. And it forms a very interesting shape, which I will draw poorly, but it's actually less accurate than the book's picture, but I think it makes a little more sense. Yes, Sarah?
Aren't the ribosomes made in the nucleolus?
They absolutely are.
So then why don't they just get why do they need, if they already are in the nucleus at one point, why do they have?
Okay, so amino acids are actually acidic, and we don't want to bring them into the nucleus, right? It's kind of like I may have my blueprints in a library, but I'm certainly not going to show up to the library with all my wood and labor and construction workers and work there. That's not what that space is for, right? So I'm going to do the job outside where I won't mess up everything.
Okay.
Does that make sense? It kind of works like that. Okay, this is a terrible drawing. It doesn't really look like this. It's not in the shape of a cross, it's just how I'm drawing it, because it gives you an idea that it actually has these three folds coming out of it. Now, because it's RNA, it has all of its strands, all of its bases pointing to the inside, except for one spot, and it'll be very important if we talk about what its job is later. Right here, these bases point outside. So let's say we have A, C, U right here, random letters. Because they're pointing outside, what can they hold on to? Yeah, something else, right? And that's going to be very important. In fact, you know it's going to stick on something that says what?
C,
Well,
C, G,
Wait, no,
oh,
wait,
we're in
wait,
RNA.
what?
U.
So it'd be-
U,
G,
G, A, right? That's going to attach on to UGA, right? Now, I'll talk about what that is a little bit later. But these are the three, and they're very important. We'll talk about what they're called later. They're very important here. Now, that's tRNA, but I've got to transport something. Remember, I have the blueprints. What do I need now to build something? Tools, sure. But you show up the blueprints, all the tools, all the workers in the world, what are you going to need? Materials, right? I need the stuff. And you're right, Margaret, and you're right. Just like Margaret said, we need the stuff to build with, right? Now, what are the pieces of proteins called? Amino acids. So what's it going to transport?
Amino acids.
Each tRNA will carry one amino acid.
Looks
They're
a
like the
snack.
trucks here. They're going to deliver our supplies. They're going to deliver our building materials. tRNA carries amino acid. Now, also to the ribosomes, right? Because if they don't, there's no instructions. So they're going to go to the ribosomes as well. Okay. The last type of RNA is the rRNA, and it stands for ribosomal RNA. If you had to take a wild stab, what do you think it is? It's a ribosome, right? rRNA makes the ribosome, and instead of being a weird T shape or a straight line, it forms a great big blob. It's called a globular RNA.
A globular.
One big glob, one littler glob. Unless you learn what those globs are called, probably tomorrow.
Is it really called globular?
Globular RNA. That's the exact name of it. In fact, proteins that shape like this are called globular proteins. So, this is globular RNA. It's going to form a big blob, and it's the site of protein production. So it's kind of both the work site and the workmen in a certain sense. It's going to kind of pull, it's where the proteins are made, okay? And if you remember, where do most ribosomes tend to hang out?
That little,
Like,
They're bumpy.
The rough.
Rough
Endoplasmic
rough
reticulum.
That's right, rough endoplasmic reticulum. Not all of them are there, but a lot of them are. So, that's where the ribosomes are. So to run through these quickly, messenger RNA does what? From to tRNA brings amino acids to the ribosomes. rRNA
Makes the.
is the ribosomes, right? It's the site where everything's made. So rRNA is the ribosomes.
Oh, that makes sense.
Now, all of these, well, I mean, it makes up, they're made out of it, right? Ribosomes are made out of rRNA.
Oh.
Now, all this RNA, all that DNA code is to make what? The whole purpose is to make what? Protein. So this process, we're going to cover how it works. It's called protein synthesis, the making of protein. Protein synthesis, the making of protein, okay? And since proteins are the building blocks of life, in one sense, this is the process that allows for life. I know it's strange to think of this, but everything you do relies on you making proteins that do that thing, okay? When you get excited, it's because there are different proteins. When you are full, it's because there are different proteins. When you break down food, it's because there are different proteins. When you hit someone, it's because there are different proteins. And when you go to sleep, it's because you've been slowly making different proteins, okay? So proteins are the things that do stuff in your body. So if you ever do stuff, that stuff is done by the proteins. Does that make sense to everybody? Okay. So this is very important. Now we're going to divide protein synthesis into two different steps. The first step is transcription, and the second step is translation. If you notice, both of those words are about what? Writing. They are both about writing, and actually one's about writing, one's about reading. But we're going to get to it later. I just want you to know both steps.
I found it.
So it's after, I think it's on the next page. After the huge chart, which you won't have to memorize, but you will learn to read. Okay, so don't panic about the huge chart yet.
I can read that. AC.
Yes, I mean, you know what it means. Okay.
I can read that.
I can read letters. I can spell words. I'm good at this. All right, protein synthesis, transcription, and translation. We're going to first take a look at transcription. Now, when you transcribe something, when you transcribe notes that I say, all you're doing is writing them down. So transcription is just changing codes. Transcription is just changing codes. If
Wait, codes?
codes.
Oh, okay.
So if I'm talking, that's a sound code. What are you doing to my sound code?
Making it written.
You're turning it into a written code, right? You're turning it into a physical code, an ink code or a lead code, whatever you want to think of it. It's just changing one code to another, okay? And in transcription, what we're going to do, what's the code we start with?
DNA.
DNA. What code are we going to turn it into? What kind of RNA? No, we're not there yet.
Ribosomal?
Not yet. We're going to, so which one of our RNA? Messenger RNA. We're going to turn the DNA code into mRNA code. Because remember, messenger RNA is the one that's all about the meaning, it's all about the information. Okay? DNA to mRNA, that's basically what's happening in transcription. Okay? Now, the way that it does this, and there's not really any awesome pictures of this in your book, but I have a few videos, one of which you will be shocked by and think is hilarious about this process happening. I know you won't believe it now, but trust me. And we'll watch it once we're done with all this. But transcription is going to occur when a certain enzyme comes in, and this is called RNA polymerase. Why do you know RNA polymerase is an enzyme? A's. Ends with A's. Very good. So,
Okay.
and just from its name, what does poly mean?
Many.
And RNA, what's it going to do? It's going to make many RNA. It's going to make a lot of RNA. No, it is an enzyme. It's going to make a lot of RNA. Okay? RNA polymerase is going to begin the process. Okay? It's going to pick a gene on the DNA, pull the DNA apart, and start reading, or rather transcribing the gene, okay? It's going to start transcribing the gene. So I want you to look at the picture I've given you right here. For the moment, ignore the sense and anti-sense strand part, okay? I know it's a little confusing, but that's more than we're going to care about here. Everybody see the RNA polymerase? Okay. once I've opened the DNA, see how the DNA is open there? Our first DNA letters are A, T, G. Everybody see that? So what do we have below it? U, A, C. In fact, you'll notice each one of these will line up with its letter. Because what's the top strand made out of? What's the bottom strand made out of? So that's how I turn DNA into RNA. Does that make
By
sense?
disconnecting them?
Yeah. Except polymerase is making sure that DNA doesn't get put on there. RNA does. Okay? So, the polymerase is going to line this up, and as it moves along the line, it's going to form a long line of RNA that matches the DNA. Okay? A long line of RNA that matches the DNA. That's what the polymerase does. It's going to make a long line of RNA that matches the DNA. Now, what's interesting is that this language has words. In fact, all the words in this language are 3 letters long, which means they don't have a ton of words, but they still have enough. And these 3-letter words are called codons. Codons are 3 nucleotide-based words, and they form up the language of RNA. mRNA's language is in codons, 3 letters for each.
Wait, 3 nucleotide what?
Words.
That form
Why not?
RNA.
You can say letters. So mRNA language, all the words are 3 letters long. Okay? All the words are 3 letters long.
These are the codons.
And they're called codons, right? The 3-letter words are called codons. Now, the first codon of every single sentence, in fact, this codon means start, is AUG. AUG is going to be our first codon. Now, if you'll notice on our example codon here, it's a little bit off. We're ignoring that for right now. It's just giving you an example. But our AUG codon is going to be our start codon. And it codes for something called methionine. That's what the MET means right here. It means methionine. And methionine is actually going to be our very first amino acid, excuse me. It's going to be our very first amino acid is going to be our AUG right here.
I don't understand the methionine.
So
Where is that?
each codon will code for, well, that's the MET thing, so it's right there. See it next to AUG?
Oh, there.
That's methionine, and we'll look at, in fact, if you look at the chart, you'll see that AUG is going to make what on the chart? If you can track it down. Methionine, or start. So it's just going to be our
Oh, I
first.
found it right there.
Good. Now, what's going to tell us to stop?
UGA,
UGA or
UAA, UAG.
UAG. I always remember UGA because everyone can stop
Georgia.
Georgia.
Wait, what is the
What
did she
have
say?
to do with it?
That's
what I was thinking.
It's up to you to stop ugly. Only you can prevent. All right, so like the Smoky Bear. Of,
Except Auburn.
Hey, Smoky Bear. UGA is Georgia.
I know. Didn't Auburn beat Georgia?
No,
No,
Georgia
that's
killed
right.
Auburn.
Yeah.
That's what I
This is
mean.
a joke. This is a joke.
So,
It's not a law. I'm not teaching you a biological fact. I'm just saying that's how I remember it.
Or ugly, remember ugly.
yeah, I can do that.
Hey, don't talk to her like that. Oh, I was saying remember. She said it. So we have, Okay, so we have these stop codons, and the stop codons are going to stop the protein. So the protein will be made from methionine until we hit a UGA, a UAA, or a UAG, at which point the process will stop. It will cease function. So let's take a quick look at that chart. I know it's scary. On the next page.
Got my name.
We got it.
If I have, so let's try to figure out how this works. If I have CUU, the first thing I'm going to do is find C on the left side. Everybody see that?
Yes.
Then I find U at the top, right? So what will CUU give me?
Leucine.
Leucine. So if I tell you I have the codon CUU, and I say, what amino acid will I make? You would write leucine. Okay. What if I had AAU? I start in the A on the left. Everyone see that?
Stop.
I see the A at the top,
Asparagine.
asparagine,
Yeah.
which can be found in.
Wait.
Asparagus.
Yeah. All right, your homework for tonight is 293, 1 through 5.
Wait, so the bolded is the answer that it makes.
Wait.
So the
UAA
bold things are the
is
amino
stop.
acids.
Yeah, wait, no, not AU.
Those aren't
AU.
A.
You.
Isabel,
Yeah.
did you ever find out why Mr.
Oh, that's
Johnson was saying